Science on Surfaces. A bigger perspective on the small.
Welcome to this podcast, Science on Surfaces, a bigger perspective on the small, where we zoom in on science that impacts our everyday lives. Today we'll talk about tissue engineering. We'll talk about what it is, how it can be used, where surfaces come into play, and more. Here in the studio I have Professor Gatenholm, Professor of Biopolymer Technology at the Department of Chemistry and Chemical Engineering at Chalmers University of Technology. And I also have Professor Bengt Kasemo, Professor of Physics at Chalmers University of Technology. Both of you very welcome.
Thank you.
Thank you.
So as always, let's start with the basics. Tissue engineering, what is that?
Tissue engineering is a technology which enable to make tissue. Very simple.
And how does it work?
How does it work? It is it utilizes cells. So normally we would be working with human cells. Utilize the scaffolds, which are the temporary extracellular matrices, which will keep the cells around. They will guide the cells, they give support. And the growth factors, which are necessary, for example, for cell differentiation, but also to provide the cell different signals.
And would you say scaffold? What is a scaffold?
Well, a scaffold, when you build the house, you will put the scaffold around the house. So we expect that it is something what should be taken down during the tissue engineering process. But we have some examples of permanent scaffolds which stay after building process. But most of the scaffolds are the polymers.
Okay.
So no metals, some ceramics, but mostly the polymers. And some of them could be biopolymers, some can be synthetic polymers.
And this is the structure guiding the cells into how they will grow, like a structure they will grow into.
Exactly. This is a structure, and it is ideally when the cells which produce the extracellular matrix. So take example the cartilage or the skin cells, fibroblasts which produce the skin tissue. So they will produce extracellular matrix, but it will take a little time before they will do it. So they need to have a place to be before they start to produce extracellular matrix. Later on, when they produce extracellular matrix, the scaffold should disappear. So it should be preferentially biodegradable. Ideally, should have a slow degradation time to slowly disappear when the extracellular matrix is produced by the cells.
So the extracellular matrix is like the scaffold. It replaces the scaffold.
Exactly. So scaffold it's an artificial extracellular matrix.
Okay.
This is very simplified because you have many cells which do not produce extracellular matrix. So cardiomyocytes, they are beating cells, or you have cells which you have neural cells which connect to the neural network. You have hepatocytes or insulin producing cells like Langerhans islands. These cells are bioreactors, so they will never produce extracellular matrix. They need some housing. So that scaffold will be more packaging system which need to be vascularized.
Okay. Many different words here.
Many different words.
One thing, or difficult words, one thing at a time. So you said if they don't have the extracellular matrix, difficult word, then you need some sort of container for them to grow in.
Yes.
Right. To create the structure.
To keep together. Yeah. Exactly.
And then, so what kind of tissue can you grow like this?
Well, you can. What has been grown successful, it is cartilage tissue, skin tissue, bone tissue. But most of the tissue have been grown really. So we in the last 20 years been tremendous progress on the cell culturing, learning how to culture the cells, different scaffold materials, also access to growth factors and the media which you can buy today. So it has been a lot of progress. It's much easier to do it today than it was maybe 20 years ago.
Okay.
a common denominator of the tissues you mentioned is that none of them need vessels. They don't need vascularization.
It's correct.
So if we move now into vascularized tissue, how much more difficult is it to grow those tissues, like liver or kidney?
Exactly. My point is really that from growing tissue to growing organ, but even tissue which required vascularization, it is a challenge,
and this means then blood vessels through the tissue.
Through the tissue, and when it's not only one blood vessel, it's you know, for hepatocytes, so for the liver, or for we work with pancreas now for diabetes treatment. It is every cell need really the access to a lot of oxygen and nutrients. So it's not a one vessel. It is this whole vascular tree with a small capillaries. So you can envision those cells like the grapes sitting on the stalk. Mm-hmm. And that is much more challenging to do to grow a vascular tissue. And so when you move really to the organs, because the liver is an organ, the kidney is an organ, but even the skin is highly vascularized, and those earlier skin models which have been grown by tissue engineering have not been that successful. So now the new models are coming with vascularization. And that is a challenge, of course, for the engineer, because now you need to handle several type of cells at the same time, but also to engineer that. You need to grow the vascular tree within the growing tissue.
But this is being done.
This is
successfully
currently being done. I mean, just very recently.
Yeah, okay.
We see almost every week some great papers coming out.
But it's at a research level, so it's not implemented.
It is on the research level, but interestingly, when you implant the tissue, you see in many cases the spontaneous vascularization. So in surgeons know that. I mean, if they transplant the skin, they will put some connection and can really promote vascularization process. So I think we are in quite early. It's a research very early stage of understanding vascularization, and you know that. Blood vessels grow spontaneously. I mean, that's really the problem with a cancer, that the cancer cells are recruiting the capillaries in you have a sprouting system when the endothelial cells go to the cancer, to the tumor, and they provide a lot of nutrients, and that's really getting the tumor growing. So we know that the growth of vascular system can be spontaneous, but we need to really learn how to mimic that.
How well are the key factors known that induce vascularization and guide the vascular? How well are they known, and which are they?
You know, with the growth factors, VEGF are very important. Cell cross-talking, but very often you need to have a pericytes, endothelial cells, but also some smooth muscle cells, because the only migration of the cells is not good enough to making the vessels. It have to follow up by the self-assembly process and making the tubular structure. So this is what we are. You know, a lot of things in tissue engineering getting back to the development biology, because everything, all the organs, tissue, they start to grow. And so when I teach tissue engineering, always I hire the professor from development biology from Gatenholm University, and he gives a crash course on the development biology, because that is really. If you see the embryo formation and the growth of all the tissue and organs are growing, it's the time, but we're starting from two cells, and this is really that process which the tissue engineer needs to understand and mimic. provide the environment for the cells to become a tissue, to become a organ. So
when you bring in development biology and embryology as good starting points to learn about the vascularization, you I guess inevitably also think about stem cells. So how important or potentially valuable are stem cells in this vascularization challenge?
I would say generally the stem cells discovery, but also the technology of stem cells have been extremely important for tissue engineering, because one of the challenges is a cell number. You need to have a lot of cells if you want to produce tissue or organ. And during the so what I've started with isolation of primary cells, and then to expand them. And during expansion, many cells they will change the phenotype. So they will, when we grow the chondrocytes and we double them, and they will become fibroblasts. So it is only limited amount of passages which you can use. And when you don't have enough cells, now the stem cells can be expanded unlimited. You can produce billions of cells, and they are still the same. So really the stem cells make the tissue engineering a lot of progress. Now we still have the next step is induced pluripotent stem cells, which is because even in stem cells, you have an access. Okay, so you get a bone marrow, you need to have a donor, you get your dose of a bone marrow stem cells, you can expand them many times, but still you need to have access to those cells. Yeah. You can work with adipose-derived stem cells, but you need to get a liposuction of somebody, it's a donor related and so on. Where induced pluripotent stem cells, which the technology is quite new, it started with Yamanaka, Gordon, Sir Gordon, not many years Nobel Prize.
You mean the ability to back off mature cells into stem cells?
Exactly. And then this really, it's another big opening, because now with induced pluripotent stem cells, the scientists are learning now how to differentiate them to different cell lineages. So with stem cells, and particularly induced pluripotent stem cells, we have unlimited access to cells, which is a key for tissue engineering. Now we need to have scaffold materials, and I am biomaterial person, and I would say that it's the progress has not been that big as it could be. Everything started with Bob Langer, really, from MIT, and he had different polylactase, polyglycolic acids, or PLA, PGA copolymers, which fortunately were tested for the drug delivery in the 70s. So when he started his first experiment, he took those polymers from the shelf and said, well, let's try to put this with some cells, and they are biodegradable. And it worked. He could show that the tissue, it was the first ear on the back of the mouse was grown, published a paper with Joseph Vacanti. Those polymers stayed for many, many years because they were FDA approved, they had a good records. If they have a bad, maybe not. And then we've seen tremendous progress of creative chemistry of making different polymers, but regulatory aspects are really make it difficult to bring those polymers to clinic. So if we're interested in the translation, to bring the tissue engineering to clinic, to use those tissue to replace tissue for regenerative medicine, that is really the limitation.
So how important is this surface interaction between the cells or biomolecules and the scaffold, for example? How important is it for the success of the growth of the tissue?
It is very important, and it is really the we have different stages in the tissue growth. So if you think about it now, that you start with fibroblasts, or let's get a fibroblast, very simple cells, which supposed to make, we're going to make a skin, and you put them into the alginate, which is a good polymer. They don't recognize the alginate molecules. They will never stretch. They will never migrate. They stay as spheres. If you take now RGD modified alginate, so you put some sequences of amino acids which are similar to fibronectin, and this is a polymer which fibroblasts liked, you will see dramatic effect of a cell behavior. Now a cell started to stretch, and they start to adhere, and they can start to walk, they can migrate. So it's very important. Now you don't want to have extremely strong adhesion, because if you get a cell stuck, they will not be able to migrate, they will be not able to proliferate. So it's really the balance of different interactions for different applications.
So do we know how is the interaction between the tissue to be grown and the scaffold? Is this something that you're looking into at all? You, maybe?
No, I don't think I know the answer to that. But the migration, the example you gave with fibroblasts,
yes,
tells how important the extracellular matrix, all the surfaces for that matter, are for the development of the cells. So how far have you come in that area to provide the right balance, not too much adhesion, but also sufficient adhesion for migration? How far do you think you have? Come?
I think that is it is kind of moving target where people are learning. I think good example is use of laminin. It's a Swedish company, Biolamina, which has been very successful on providing. Those molecules are crucial to coat the plates to get the stem cells to adhere. Without that, we cannot grow the stem cells. So this is a good example. Recently, one of another Swedish startups work with silk, Spider, I think it's a name. So artificial silk, combine the laminin molecules on their scaffolds. So now providing the 3D environment. So I think we are in a relative early stage of understanding the interaction, but also in early stage of providing those molecules, those Q molecules on the right scaffold and ideally we would like to have a gradient surfaces, like the gradient surfaces in physical chemistry. You remember but gradient surfaces of interactions, because if you think about it, you want to attract the cells. They're in the different stages. So if you can provide the gradient and attract the cells to migrate, but also having the different stages of adhesion.
Mm-hmm.
Yes, we talked at that time. We talked about program surfaces with
gradients,
but also putting in time as a dimension. That surfaces could be time programmed to expose different functional groups at different times in the evolution.
And we haven't really. We are in the beginning of this of the age of designing the. There is really two group of scaffolds. One would be the synthetic polymers, which have been not very much developed, but another one would be the extracellular matrix, taking from animals, taking from human, and they are very complex. So what I think it's new is the modification of a synthetic scaffolds with extracellular matrix molecules. So with different coupling chemistry, to to modify it with laminin, to modify it with fibronectin, to modify it with RGD sequences, because it's not only the question about adhesion, you can actually differentiate the cells. So instead of adding very expensive growth factors, you can think about some interaction can be surface interactions. So the surface can attach and get a right signal and start to differentiate. Another aspect of interaction is also physical interaction, because we know that cells do a lot of sensing. I mean, they sense the surfaces. And quite recently, the scientists start to understand about the soft and hard surfaces. So the cell can really sense the scaffold, the properties of biopolymer, and say, oh, this is a soft one, I'm going to become the cartilage, or this is a hard surface, I will become a bone cell. So this is also very, very interesting that scientists start to provide, and this has been quite simple experiments to making silicons with different cross-linking densities, saying, okay, let's go from soft to harder, and we see now that the same conditions where the stem cell become either cartilage, producing cartilage, or producing a bone. So
the scaffold properties are very important.
Scaffolds are very important because they are this artificial tissue before the tissue is formed. So they have to mimic the environment they need to provide the water. I mean, that's the reason why we have very often the gels, and also mechanical support can provide the different molecules to communicate. So scaffolds are a key. My favorite scaffold is really hyaluronic acid. I like very much that molecule because you see it where the life is starting. It's really it's the first extracellular polymer which cells are really producing. And the reason they do it, it's such a highly viscous polymer. It is a huge polymer which have a very high molecular weight and extremely viscous. So it is a gel which almost
captures a lot of water, right?
It captures a lot of water, but also it's almost like a solid. So by having that molecule around, the cell have already support. So
So if you compare growing tissue using the scaffold that you're talking about, if you compare that to the 3D bioprinting, that's a completely different process.
Yes. So if we look at now on the tissue engineering, how it developed and where it really stopped was a lack of ability of good seeding. So we can make the scaffold and say, okay, we need to make it porous. We make the kind of a sponge material. Now we need to seed the cells, and producing the good interconnectivity has been very difficult. With different processes, the cells don't go everywhere. So we have those sponges, and when we put the cells in on the top of them, and we think that they should come in. Now, in 3D bioprinting, you're starting with a bottom-up production, layer by layer. So what you do is that you mix what we call a bioink. We mix the scaffold, which is in this case will be the gel, liquid, together with the cells.
Okay.
So you produce now you place the cells where you want to have them, and you put your scaffold around that.
Okay.
So, and that is a huge difference, because now you end up with the cell-seeded scaffold by definition. And in addition to that, you can control. So you have to envision that 3D bioprinting is a robot, which have a very good accuracy. So we have an XYZ resolution of 10 micron, so we can place every cell where we want to have them. So if you understand exactly the morphology of your tissue, and you can get the information from confocal microscopy, you know how many cells are, where they are, you can program that robot now to replicate that. And you can say, okay, those, and now you can do many cells. We see that the new printers have not only 3 heads, they have 6 heads, they have 9 heads. So every head, it's a syringe mixed with the cells with a hydrogel. So you can
do you have different cells in each of those?
You can have different cells. You can have different biomaterials. You can have biomaterials with different growth factors. So you can produce now the microenvironments, not like we were doing with scaffolds. It was one scaffold, spongy, one cell of type, type of cells which couldn't go in scaffold. Now we can actually produce everything. So that is really the promise of 3D bioprinting, where the technology which allow you to bottom up produce layer by layer cell seeded scaffold from the beginning, and also reproducible, because every time robot do the same things. And I think a very big advantage, many cells at the same time, many cell type at the same time. So you can come with one head, you put endothelial cells, you come with other head, you bring the smooth muscle cells, you can put the fibroblasts, you can put the keratinocytes, we put the melanocytes. So when we do the model of skin, you can already make the pigmented skin. You can do the cancer model. You can print the tumor, even if you don't want to make a tumor, but you want to understand how to treat the cancer. So when you can do the replica of the tissue and organ.
So, but these two methods, are they both routinely used to create tissue that is being used on people, so to say?
I would say that I wouldn't call them two different methods. I would say that. But everything is tissue engineering. 3D bioprinting it's a tool.
It's a tool to
It's a tool to making the tissue if you want to. I think we should then differentiate between the use of a tissue and organ in regenerative medicine and then as a models. So as a models, I think there's been tremendous progress to produce the small organoids, small organs, or pieces of tissue. I can give an example, the drug screening, of course, but also the models of skin, when all the cosmetic companies are not allowed to test the cosmetics on the animals. So now all the companies producing their own human skin models, and that you can do it with 3D bioprinting. When it comes to testing the drugs, microfluidics made a lot of progress into the organ on a chip, and it is technology which is much more mature than 3D bioprinting. But it was very often on the single cell level. You can put cells and you can expose them a drug. Now the scientists start to fuse those technologies, the microfluidic technologies, 3D bioprinting technologies. So you still are interesting. I think microfluidic have very smart, many smart solutions.
Okay.
So you have your channels, you have the chips, and you can put different models of. You can have a heart, you can have lungs, connecting them. But now you can do it in 3D. You can make a small 3D lungs and make a small 3D heart model and still connect them. So this is really hot to produce the human on the plate now, not only on the chip, the different organs.
When you talk about the 3D printing and compare it to another way of approaching making an organ, you can almost discover two paradigms. One is that you use 3D printing, as you described, to make something that is far ahead, almost ready for use, and very heterogeneous, different parts of the scaffold doing different tissue and so on. And then you implant it for regenerative purposes. That's one approach. Another approach is to say, I take something very simple with stem cells and I plant it and I let the body do the work. Can you see these two lines of development as competitive, or or more as complementary and applying to different types of tissues? It would be interesting to see how you view these two development lines.
So you are bringing a very, very important issue of implantation, transplantation, and in various, I think, tissue engineering have not been that successful in translation. We don't see that many tissue and organs which have been produced with this technology, and probably because that we in tissue engineering was very much focused on in vitro studies and develop a bioreactor, make it with tissue and organ ready in the lab, and then a failure in bringing it to the clinic in translation due to rejection because of biomaterial questions or regulatory aspects and so on. I think that printing, for example, or with tissue engineering technology, creating the scaffolds with stem cells and implanting, it's very attractive because the body have a ability to differentiate those cells, and so I think it's a good. There is one of the challenges are mechanical properties, because if you think about a cartilage, which is important for osteoarthritis, it's a big failure of our design, because the cartilage is not renewing. It's have no vessels, have no neural cells, so one destroyed, never repair. Now there is a technology developed here in Göteborg by Lasse Pettersson, Mats Bridberg, and Anders Lindahl of autologous chondrocyte transplantation, which has been is actually the first cell therapy which is in clinic 30 years old already, taking the biopsy, growing the cells, expanding and injecting back. But in completely destroyed cartilage, it is almost impossible, because there is no place for those cells to grow. You can inject those cells in the small lesions. So it is really bringing the challenge of creating the materials, the bioreactors inside the body. Some of the scientists do with banking. I think the people in Finland have done to start with for jaw, for example, which have a lot of mechanical forces. So you don't want to make the bone an implant. So they put the stem cells together with biomaterial and they put it under the skin. So we call it banking. Okay. So they grow there and they differentiate without that mechanical forces. And then they take them out and put in the jaw.
So it grows in the body.
In the body,
but
in the patient.
In the wrong place, though.
In the wrong place, but in the right body.
Yeah.
So you get your differentiations, you get all the reaction, and so this is kind of, I would say from technical point of view, I mean it's 2 surgeries, probably cost and so on, but we are experimenting. You know, it says everything. I think if it's helping the patient, I think it should be tested.
There are many different development, or rather many different applications of tissue that has been engineered. And so if you are, I would like to ask you how hopeful you are for different applications that you see down the line. For example, regenerative medicine to replace organs is of course a dream. But even if that fails, there are other development lines, for example, to make tissue to evaluate, screen drugs, which can save animal experiments and so on. So there is recently a new course and initiative at Exeter on that particular issue. And in addition to that, you can say that if you have engineered tissue, you can develop diagnostic tools, ex vivo and so on. Would you be able to rank or comment on the different potentials, different hopes for these different development lines. I could add the more exotic development line of artificial meat as a yet
exactly.
another one. So there are many different applications, and if one fails, you may be successful with the other.
I would say, firstly, from a regenerative medicine point of view, one tissue organ which has been successful is the bone. I mean, been tremendous progress on the bone tissue engineering, but the bone is so nice because it's healed itself.
Yeah.
So you know, whatever you do, you will be successful. But people have been helping the patients with larger bone deficits, you know, adding the biomaterials together with cells. I think that there's a lot of cells recruitment also there. So this is a good example when tissue engineering has succeeded for bone tissue engineering for repair the bone. Skin has been tough, and it's very successful as a models. It's been spreading out for the drug testing, but also testing cosmetics. But we haven't been able to produce what really is necessary for the burn patients to quickly produce the patient's own skin or skin which will not be rejected, because you know those patients need within few hours a large amount of skin, and that is one of a really important part. I would comment for you about for the drug discovery, it's enormous. Discovery and screening is enormous tool, because it makes such a process but such shorter and more efficient. Not only avoiding animal models, but honestly speaking, most animals models not always work, and there's so many different animals which drug company have to move from mice to hamster and rabbit and organ.
So to have human cells would be a great disadvantage.
And in addition to that, you can do this personalized medicine, because you can have if not every patient, a group of patients. By different races, different people with different genetics prerequisites, you can take those cells and say this is that group diabetes patients. We can show how the skin or chronic wounds can heal in a diabetic patient, because we can do that model. Something what excites me a lot is it's a space potential of tissue engineering with space, and we are involved in a small. It's at this stage a small program, but we hope it will grow. So as you know, probably on the space shuttles, they are already printers to repair the parts. You know, part of the space, the space shuttle. Yeah. But the scientists realized that for the long travel, you need to also have the tissue bank, the cell bank. You need to repair not only astronauts, but people who will live somewhere far away. So both NASA and the European Space Agency, ESA, has started a program on tissue engineering, and particularly 3D bioprinting. So there is a NASA challenge called vascularized tissue, and we are part of that with Florida Institute of Technology. There are many teams, and it is a competition between the. So you can make your team if you want, and you can sign up, and if you. And there is it's kind of it's very American way. It's very well defined. It has to be 1 cubic centimeter tissue with well defined size. So like it's liver, or is it heart, or is it pancreas, which has to be vascularized, I think, to 70% and be active in at least 1 month. So it is, and if your team can do it, and you will get $500,000 the prize. It's interesting because it's kind of bringing and this is we come to this bottlenecks and challenges: vascularization. Vascularization, because if you really want to make more complex tissue, it's always vascular.
So how far in the future is this, you think, for someone to succeed and win the prize?
It's the deadline was this September, and it has been extended another year because nobody could make it. And the teams, it's a Harvard team, it's a Stanford team, it's an MIT team, it's a top scientist. I think that, honestly speaking, I don't, maybe another few years, but at least there is a target, the focus target. Say, okay, make that tissue with that amount of blood vessels, show it works, It's kind of it's a quantitative and qualitative target. Vascularization, it's very important. It's difficult, and there I think the surface interaction will play a very important role. We are working in our lab making some, and we can print tubes. We can print the vascular trees, but it is a challenge to getting the endothelial cells to line up inside in those artificial vascular systems, and they are quite. It's a lot of interaction between endothelial cells and the surfaces. So we like fibronectin, maybe they like some laminin. So I think this is a field, but maybe we will go back to QCM for screening different surface treatment, different molecules, because there are so many possibilities. If you're thinking now about bioinks, I mean, you can mix anything with bioink, but you want to have interaction of endothelial cells, because when we talk about vascularization, it's not only the channels, it's not only the tree. It is a lumen which is lined up with endothelial cells, and they have to be alive. So they need to be very happy.
So thinking about this challenge, then it sounds then like organs are still not impossible, will not be impossible to make, all right?
That's what we are hoping, because without that, the whole, I think it would be the failure for technology not going for organs.
Yeah.
So when you need to understand, okay, to making the organs, we need to have a vascular tree, we need to have vascularization, we need to build it. We need to get the cells to grow into that. We need to give them nutrients. We need to provide them oxygen. We need to have a perfusion system. So it's another technical challenge, but I think it's very focused now, because it has been kind of a little naive from a tissue engineering point of view, that we haven't really taken into account the needs for vascularization for many years. In people have been growing cells, and okay, I have a piece of tissue, and then what? So this is, I think, important.
Well, I think that if you have time for one more question. My impression of the field is that some of these areas will be successful, and you pointed out the regulatory aspects as very important, defining a timeline. So
yes.
Maybe it takes a long time to get real regenerative medicine of organs. But that development will benefit from the ones which, in my view, are nearer in time, like drug screening.
Yes.
Maybe also, and one thing we haven't commented on is on diagnostic tools.
Yes.
So how valuable do you think engineered tissue can be for developing diagnostic tools? It could be both imaging and chemical diagnostic.
Extremely valuable, because it's so. I mean, you cannot really develop diagnostic tools without having access to living tissue. So that's very important. I want to tell a little about at the end of the Tissue Engineering and Regenerative Medicine Association. So there is a what's called the term is Tissue Engineering and Regenerative Medicine International Society. And I'm going on Sunday for the meeting to US. It's going to be in Orlando, and it's have 3 chapters. It's Asia, Europe, and America, and each one have about 1,000 members. So both conferences are about 1,000 people, and every third year it's a World Congress. which bring almost 3 chapters. So it's more than 3,000 people. But what is exciting to see, it's so young field. It's and it's so it's such a mixture of it's. I think it's not enough surgeons and clinicians. It's a small group, but it's growing. But they're business people, they are biomaterial people, they're transplantation people, they're cell biologists, and all people are very young. So it's kind of, I would say, when you look at on the poster session, average probably 25, 30. And it's a growing field, It's at this conference probably going to be 1,000 posters, and so many PhD students, and they seems to get jobs. Everybody, so it's kind of interesting to see that the tissue engineering, as you mentioned yourself, as a technology field, started to produce the tissue and organ to treat the patients, but it found so many other applications for diagnostics, drug screening, drug delivery, and so on. So the tools which are developed are finding so many applications and creating many jobs.
So we can expect a lot to happen in the future, then I guess, based on what you're saying now.
Absolutely.
With all that interest.
Wish you good luck.
Thank you so much. Thank you.
Yes, I think that's all we had for today. That was a great introduction to the topic of tissue engineering. So thank you for listening to this episode of Science on Surfaces, a bigger perspective on the small, with me, Malin Edvardsson, Paul Gatenholm, Professor of Biopolymer Technology at the Department of Chemistry and Chemical Engineering, and Bengt Kasemo, Professor of Physics, both of you from Chalmers University of Technology. Thank you.
Thank you so much.
And I would also like to take the opportunity to mention to those who are listening or watching that if you're interested in surface science and related topics, you should check out our blog, the Surface Science blog. Thank you.
Thank you.
All right.